I'm Suaid Farid Hafiz, Professor of Medical Microbiology and Immunology. Today, our topic is HIV infections and AIDS. I will be covering HIV, the causative agent of AIDS, its life cycle, pathogenesis, and laboratory diagnosis. Human immunodeficiency virus, or HIV, is the etiological agent of acquired immunodeficiency syndrome. It is a non-oncogenic RNA virus from the family Retroviridae genus lentivirus. There are two types of HIV, type 1 and type 2. Both types are further divided into groups. Each group corresponds to distinct lineage independently introduced to the human population from the non-human primates. The source of HIV type 1 is chimpanzees and gorillas, while that of HIV 2 is the Suti-Mangabe endangered West African primate. HIV-1 and HIV-2 both can cause AIDS. HIV-1 accounts for 95% of all HIV infections worldwide. On the other hand, the relatively uncommon HIV-2 virus is concentrated in West Africa and in countries with links to West Africa. HIV-2 is more than 55% genetically distinct from HIV-1. It is less infectious, progresses more slowly than HIV-1, resulting in fewer deaths. However, without treatment, most people living with HIV-2 will eventually progress to AIDS and die from the disease. HIV-1 represents an extraordinary degree of genetic diversity resulting from the error-prone replication cycle associated with lots of mutations and even hypermutations. The most predominant HIV group is Group M. Group M is responsible for more than 90% of the global HIV pandemic. It is further divided due to genetic diversity into 10 subtypes, and the subtypes are even, some of them, further divided in subtypes. There are also what we see as, we term as circulating recombinant forms, and these result from recombination between different subtypes, resulting into hybrid virus that we refer to as CRFs, or circulating recombinant forms. HIV is spherical envelope virus, icosahedral symmetry. It possesses external spikes formed by envelope glycoproteins GP120 and GP41. It is also studied by host cell proteins. Inside the virus envelope is a layer called the matrix, and a dense cone-shaped nucleocapsid composed of the P24 protein, and as well as two identical copies of single-stranded positive sense RNA genome. The genome is associated with two important enzymes, the reverse transcriptase and integrase. The HIV genome encodes for 3 structural genes, GAG, POL, and ANV, and 6 regulatory genes. The GAG gene codes for the P24, the viral capsid protein, the P6 and 7, the nucleocapsid proteins, and P17, the matrix protein. POL gene codes for the for the reverse transcriptase, the integrase, and protease, 3 important enzymes in the replication cycle of the virus. The reverse transcriptase has 3 enzymatic activities, three domains with 3 different enzymatic activities: the RNA-dependent DNA polymerase activity, the ribonuclease activity, and the DNA-dependent DNA polymerase activity. The integrase is essential to integrate the double-stranded DNA into the host genome, while the protease cleaves the proteins derived from GAG and POL genes to functional proteins. Regarding ANV, it is derived from the word envelope, and it calls for the envelope associated glycoproteins, or We call them GP120 and GP41. These proteins are embedded in the viral envelope and enable the virus to attach to and fuse with the target cells. Regarding the regulatory genes, we have 6 regulatory genes. TAT codes for the TAT protein that enhances the efficiency of viral transcription. REF codes for the REF protein involved in the export of unspliced and incompletely spliced messenger RNA from the nucleus to the cytoplasm. And here I would like to highlight that when we refer to the unspliced, it is the RNA that will act as genomic RNA. When we refer to the spliced by the messenger RNA, are the messenger RNA in which the introns were removed and the coding ends are tied together, joined together to be transported to the cytoplasm for translation into proteins. VIF codes for the viral infectivity factor, a protein that disrupts the antiviral activity of the infected cell and thereby ensures the cell machinery is directed towards the production of viral particles. Regarding VPR, it codes for the viral protein R, which enhances the transport of viral core from the cytoplasm into the nucleus. VPU, on the other hand, is the codes for the protein U, which enhances viral release from the cell. Regarding NEF, or the negative factor, it is the gene coding for the negative factor. This protein makes the infected cell more effective producer of the fully infectious viral particles, and at the same time less visible to the immune system through downregulation of several cell surface receptors, the CG4 and class 1 major histocompatibility complex. Actually, it was noted that deletions or mutations of NEF have been found in long-term non-progressors, which means that these viruses with NEF mutations are weak viruses or avirulent viruses, and people infected with NEF mutants, strains of the virus, are surviving the infection much better than those with the virulent forms of the virus. Coming to the life cycle of HIV, we can say that the life cycle can be divided in 2 forms: latent cycles of infection and productive cycles of infection. The first step in any viral infection is entry into the host cell or the target cell. Here there is 2 mechanisms for this entry. There is the actual mechanism through the specific viral receptors, but prior to this there is an important step for HIV, which is the viral attachment to non-specific cell surface receptors. This non-specific binding is very important, and it's very important to highlight. Why? Because it's a very important tool to help the virus to infect even susceptible or target cells, because actually it leads to concentrating of the virus on the surface and making it more capable to bind to the specific viral receptors. So this non-specific binding aids and supports and enhances the binding to the specific receptors. Other than this, a very important value of this attachment to the virus is that it will contribute to the dissemination of the virus, because by binding to non-specific receptors, this binding will stabilize the viral particles and will make the cell that held it like this capable of transporting of the virus to other susceptible cells, target cells, or even target tissues. And actually this results in viral dissemination in the human body and contributes majorly to this. For example, when the virus particles are there in the vagina, the dendritic cells can bind the virus and transport it to the regional lymph nodes, where the virus finds a huge numbers of susceptible CD4 positive cells and gives the virus a big chance to bind to these cells and replicate and produce a major progeny during the acute viral syndrome or acute viral infection. Following this multiplication, it will go through the lymphatics and to the bloodstream to seed and spread to other organs. So this type of attachment and this type of transmission utilizing the mobile cells of the dendritic cells, mobilizing the virus from the vagina and surface mucosa into the regional lymph nodes to find a pool of CD4 lymphocytes to multiply and increase in number and then disseminate into the bloodstream to reach other organs is called trans-infection, and it contributes to the viral dissemination in the human body. That's why we'd like always to highlight in this non-specific attachment. But what about the binding to the receptors? This is actually the phase by which the virus will start its replication cycle or its life cycle. So actually, the viral, it acts through the function of both the envelope glycoproteins, the GP120 and the GP41 in sequential actions. First, this GP120 binds to the CD4, resulting into conformational changes. These conformational changes makes it more capable to bind further to the co-receptors which are essential for binding, for effective binding. Another conformational change takes place, resulting in exposing of the GP41 and its power to bind and induce diffusion with the cell membrane, and this results in the entry of the virus into the susceptible cell. Once the virus enters in the susceptible cell, the reverse transcription process starts, utilizing the three enzymatic domains of the reverse transcriptase. First, the RNA-dependent DNA polymerase activity copies the HIV RNA into a complementary DNA, which we call copy DNA. At the same time and simultaneously, the copied part of the RNA becomes digested or degraded by the activity of the ribonuclease activity of the reverse transcriptase. Subsequently, once the DNA, copy DNA is formed, a linear double-stranded virus, utilizing the DNA-dependent DNA polymerase activity of the reverse transcriptase, will create a linear double-stranded DNA from, this part I will reverse. So following the binding of the virus and the release of the virus capsid into the cytoplasm, the reverse transcriptase enzyme starts its activity, transforming the positive sense single-stranded RNA virus to a copy HIV life cycle. HIV life cycle can be divided into two types of cycles: latency, a latent cycle, and a productive cycle. The first step in the life cycle of the virus is its attachment to cells. There are two types of attachment: the non-specific attachment to cell surface receptors, and this is an important step in virus in the life cycle of HIV, because actually it results in two benefits. One of it is to concentrate the virus onto the cell surface, thereby enhancing its attachment to the specific viral receptor, which is the CD4 and its co-receptors. At the same time, it helps the virus to be trapped by the non-specific receptors and stabilized on the surface, and that these particles, stabilized virus particles, can then be transported by the cells into different sites, which helps. HIV life cycle. The HIV life cycle can be considered in two forms: the latent cycle and the productive cycle. The cycle starts by viral attachment to non-specific cell surface receptors. The outcome of this binding either results into concentration of the virus at the cell surface and facilitate the virus interaction with the entry receptors or the specific receptors for entry, which is the CD4 and the co-receptors. So this is the virus. The non-specific binding enhances its attachment to the virus-specific receptors. And at the same time, this non-specific binding or non-specific attachment results into trapping of the virus, stabilizing the HIV virus at the cell surface, and thereby makes the cell that capable of transferring the virus particles to CD4 positive cells. And this replication independent mode of HIV transmission is known as trans-infection and contributes major to dissemination of the virus in the human body. This trans-infection is easily demonstrated here. When the exposed virus, exposure to HIV on the mucosal surface, there is very few CD4 cells at the mucosal surface. Dendritic cells bind and trap the virus particle, transport the virus particle to regional lymph nodes where there is a huge pool of CD4 cells. The huge pool of CD4 cells are susceptible cells for the virus, so thereby the virus can find susceptible cells to whereby it will grow and replicate and produce a major progeny. Following this replication, it will be released in the blood and disseminated to other organs. So this type of dissemination, which we call trans-infection, is a major tool by which the virus is disseminated all over the body and reaching susceptible cells everywhere. Following the binding, as we said, to non-specific receptors that enhances the binding to specific receptors, here in this picture we can see that the GP120 and the GP41 are essential for the binding and diffusion. Initially, the GP120 binds the CD4 receptor, and this binding induces conformational changes that enhances the binding and diffusion peptides to be exposed, which are actually part of the GP41. So actually conformational chains binding to co-receptors, resulting in exposure of the fusion peptide. The fusion peptide, which is a function of the GP41, enhances the fusion of the virus envelope with the cell membrane, and the fusion takes place and the viral capsid is released in the cell cytoplasm. Following the viral release in the cell cytoplasm, the reverse transcriptase enzyme starts to act to transfer the single-stranded positive sense RNA into a double-stranded DNA. It starts by forming a copy or a complementary DNA using the single-stranded RNA genome as a template, and then digestion or degradation of the viral RNA, followed by forming a double helix, linear double helix DNA. This double-stranded DNA is then transported into the nucleus. This, what we call the reverse transcription complex, is transported from the cytoplasm to the nucleus. This process is tightly associated with the uncoating process, and for a long while it was not known exactly where uncoating takes place, but recent data indicate that HIV-1 completes reverse transcription and uncoating inside the nucleus. Following the entry of the virus into the DNA virus and its uncoating in the nucleus, the integrase enzyme acts to integrate the virus DNA into the host cell genome, and from this point on it is called the provirus DNA, the provirus DNA. And following this, the provirus DNA continues to be an integral part of the cell genome throughout the lifespan of the cell. Even if the cell divides, it's divided through, it is divided, and the progeny of this cell are still carrying the provirus DNA into the other cell. So what is transcription and when does it occur? The provirus, as we said, will remain integral part of the cell genome and will continue even to replicate with the replication of the cell genome, and all the progeny of this cell will be carrying the provirus DNA, and the cell will be having this provirus DNA throughout its lifespan. So what happens to the virus? Either it stays like this, and this will be, we call it no virus expression or latency, but sometimes it passes into what we call low-level chronicity with sporadic virus expression, and in this phase usually the cell is maintained, it is not lysed, and the virus is released through budding from the cell membrane, or full-blown virus expression with production of a large number of virus progeny and lysis of the cell. The control of these states is determined by viral regulatory proteins as well as cellular regulatory proteins. The initiation of productive HIV infection requires cellular activation signals. So the cell must be activated, and this is what is commonly referred to as the cells that are acting or are induced to fight infection are the cells more prone to be destroyed by the HIV. So the more the antigenic stimuli the patient is exposed to, the more the cell depletion, the CD4 depletion through productive cycles of HIV that otherwise would have been latent inside the cell. During the productive HIV infection, the provirus DNA acts as a template for the formation of viral genomic RNA and messenger RNA by the cellular polymerase II enzyme. Following the formation of the messenger RNA, where it is unspliced to act as genomic RNA or spliced to form the different viral proteins, it is transported to the cytoplasm and then translated. Following the translation and formation of the genomic RNA and the different proteins of the virus starts the assembly process, where 2 copies of genomic RNA are recruited to the site of the viral assembly, as we see in the figure. 2 copies, 2 strands are carried to the site of assembly, where are studied the viral glycoproteins, and there they are recruited, incorporated into viral particles, and the accumulation of proteins and RNA at the plasma membrane results in what we call curving. So this, it curves up like this to start the process of budding. Finally, the process of budding results in pinching of the cytoplasm and release of the virus particle. But the virus particle, when it is released, it is not released in a mature form. It is released as immature and non-infectious form, morphologically characterized by thick layer of radially arranged, thick layer of radially arranged gag and pol precursors. Shortly after the release, the maturation takes place. The viral through the action of the viral protease enzyme, where it will digest the gag and pol precursors into the functional protein forms and give this virus its final characteristic shape of the virus. Now we covered the HIV as a virus, the HIV and the life cycle, moving from latent infections to stimulated production of viral progeny and productive cycles of infection that usually results, if it is massive production, results in virus cell lysis, or the chronic productive cycle of infection with low rates of viral progeny produced by the cell without lysis of the cell. So now we move to HIV infection, the source and the period of communicability. The only source of HIV infection is certain body fluids of HIV infected persons, which the blood, the semen, the pre-seminal fluid, the rectal fluids, the vaginal fluids, and the breast milk. A person newly infected with HIV is infectious and can transmit the virus to another person before infection is detected by laboratory tests, a phase which we call the eclipse phase. And we'll refer to this later. He remains potentially always infectious to others. Communicability is increased in case of high viral load and an ulcerative or inflammatory sexually transmitted infections, because with more ulceration, there is more tissue fluids coming out, and with the tissue fluids carrying cells from the inflammatory cells, carrying CD4 infected cells and releasing viral particles. Communicability of HIV is reduced by treatment with antiretroviral agents. What about HIV pathogenesis and immune responses? The CD4 receptors are found on the surface of a wide variety of cells, the most important of which, of course, is the CD4 T lymphocytes. CD4 is also expressed in minor extent on macrophages, some monocytes, dendritic cells, Langerhans cells, and some B cells and brain microglia. All these cells have CD4. The monocytes and macrophages play a major role in the pathogenesis of HIV. They serve as a major reservoir of HIV in the body. And at the same time, they act as a vehicle to transmit the infection across the blood-brain barrier, resulting into HIV infection. HIV infection. The source of HIV infection is certain body fluids, namely the blood, the semen, pre-seminal fluids, rectal fluids, vaginal fluids, and breast milk. A person newly infected with HIV is infectious and can transmit the virus to another person before infection is detected by laboratory tests. He remains potentially always infectious to others. Communicability is increased in case of high viral load and in the presence of ulcerative and inflammatory sexually transmitted infections. Communicability is reduced by treatment with antiretroviral agents. Regarding the pathogenesis and the immune response, the CD4 receptors are found, which is the target cell of HIV, are found on the surface of a wide variety of cells, the most important of which is CD4 T lymphocytes. CD4 is also expressed to a minor extent on macrophages, some monocytes, some dendritic cells, Langerhans cells, some B cells, and brain microglia. The monocytes and macrophages play a major role in the pathogenesis of HIV. They serve as a major reservoir of HIV in the body. And at the same time, they act as a vehicle to transmit the infection across the blood-brain barrier. They are actually the predominant cell type in the HIV-infected brain. Once in the brain, they transmit the infection to neighboring resident cells, like the microglia, the astrocytes, and possibly even other cells. They release cytokines that are toxic to the neurons and have chemotactic functions that leads to dysregulation of the blood-brain barrier and enhances thereby the infiltration of the brain with inflammatory cells. Monocytes and macrophages, infected cells, are believed to be involved in the emergence of drug resistance, the reseeding of peripheral tissues, the development of what we refer to as HIV-associated neurocognitive diseases, referred commonly to or abbreviated into HAND, HIV-associated neurocognitive diseases. The macrophage is affected actually qualitatively, which means functionally altered, but not quantitatively by HIV. So actually HIV does not result in destruction of the macrophages. Thereby its function as a phagocytic cell is impeded, but its survival still, and it is its long survival, which is very important. Macrophages are survivors types of cells. HIV and CD4 negative cells. Does HIV infect CD4 negative cells? Actually, this was found that the virus can also infect a wide range of CD4 negative cells, including renal cells, gastrointestinal epithelium. The mechanism may involve other receptors or fusion with the cell already infected with HIV, which means, for example, a macrophage gets fused with a renal cell or cells from the GI. The reservoir of HIV in the body include the resting T lymphocytes, which have, because actually have the importance, what we call a reservoir cell, it has to be with a long lifespan, a survival cell which lives for decades, or else if it is short-lived, it will die. Once the cell dies, then the virus is released. If it doesn't find a suitable cell, it will perish and be digested. The memory T cells and other survivals, the macrophages and the brain astrocytes. And we always remember that macrophages are long lifespan. They live for decades. Immune response to HIV infection. Actually, HIV infection results in eliciting both innate immune responses as well as adaptive immune responses in the form of humoral response as well as cell-mediated responses. Yet still, the capability of the immune system to eliminate HIV is impeded due to the presence of latently infected cells, the ability of the virus to undergo high levels of genomic variation, to replicate intracellularly in macrophages, and to exist sequestrated in lymphoid tissues. Regarding laboratory diagnosis of HIV infection. The objective HIV testing is transfusion, donation, and organ transplantation safety. Laboratory diagnosis of HIV infection. The objective HIV testing are transfusion, donation, and organ transplantation safety, diagnosis of HIV infection, screening in high-risk communities at least once, and even in high-risk groups, it is considered better to rescreen at least annually, to monitor disease progression and response to treatment, for surveillance programs to assess the incidence of HIV infection and patterns of spread in communities, as well as for research purposes. There are three types of HIV diagnostic tests: nucleic acid test, antigen antibody test, and antibody tests. Regarding the nucleic acid tests, these detect HIV RNA in the blood before HIV antigen and antibody can be detected. These tests are expensive. They are not usually routinely used for HIV screening unless the person had recently a high risk exposure and he needs to know as soon as possible, or possible exposure with early symptoms of HIV infection. Regarding antigen antibody tests, these tests are ELISA or immunoassay based tests that detect both HIV antibodies and antigens. The 5th generation of HIV antigen antibody immunoassay test is a major advance as it can detect and identify HIV-1 antibodies, HIV-2 antibodies, and the P24 antigen individually in one blood sample, thereby specifically giving information on which marker is positive. Regarding antibody tests, these tests detect the presence of antibodies. There is several generations of tests. The 1st and 2nd generation immunoassays were designed to detect only IgG antibodies, which referred to as IgG sensitive, and exhibits considerable variability in their sensitivity during the early infection. When we mean sensitivity of the test is its ability to detect the antibodies, how early it will detect antibodies. So the more it is sensitive, the earlier it will detect the presence of antibodies. Regarding the 3rd and 4th generation immunoassays, they detect both IgM and IgG antibodies, so actually they are referred to as IgG and IgM or IgM and IgG sensitive assays. The lab-based tests versus what we now commonly refer to as point-of-care tests. There is a great revolutionary change in the tool to test, particularly when the need of rapid diagnosis is important and the access to the patients is difficult, like in Africa, in areas where there is remotely present healthcare system and inefficient system, and people have to travel for long distance to reach for healthcare. Antigen antibody tests or antibody tests are either, as we said, instrumental or lab-based or rapid point-of-care tests. What is the difference? The lab-based tests are usually more sensitive. They use blood drawn from a vein, are generally more sensitive than point-of-care tests. The point-of-care tests are performed usually using a finger prick, whole blood, or oral secretions. They provide immediate results, but are less sensitive. Of course, now there is great revolution in the point-of-care tests with the increased interest globally, particularly in remote areas, as we said, so the sensitivity is improving every day and with new generation of point-of-care tests. So thereby, because they are less sensitive, there's a longer period for seroconversion. Seroconversion window period is much longer than the the lab-based tests. The point-of-care test and early infant diagnosis, which is actually the most important utilization of point-of-care test. The advent of point-of-care test technology is the breakthrough that created the opportunity to increase coverage for early infant diagnosis, because it is both feasible and effective to optimize national early infant diagnosis, which is a major problem in Africa. The point-of-care testing is considered an innovative investment. Why? Because more results return to caregivers, earlier ART initiation, antiretroviral treatment initiation, and potential reduction thereby in morbidity and mortality for infants with HIV. Early HIV test reactivity and terminology is very important to know. There is three main terms we have to always highlight: eclipse period, the window period, and the seroconversion window period. The eclipse period is the period in which no existing diagnostic test is capable of detecting HIV. In this phase, it is the initial period from exposure to the first positive test. It is called the eclipse period. The HIV nucleic acid test is the first test that can detect HIV following HIV acquisition. The window period. The window period is the time between acquisition of HIV and the time when the laboratory test will accurately detect HIV infection in almost all persons. The window period depends on the diagnostic test used, and it is the time period to rule out HIV infection after a potential exposure. For the CDC, which is the Center of Disease Control in US, recommends using 45 days as the window period for laboratory-based HIV 1 and 2 antigen antibody assays, and 90 days after all HIV antibody tests and all HIV point-of-care tests. So actually, this is the recommendation, that we do not feel comfortably and safe that is the patient or the exposed person did not acquire HIV except after the 45 days if we are using HIV 1 and 2 antigen antibody assays, or 90 days if we are only using antibody-based assays. What is the seroconversion window period? When we say seroconversion, it is antibodies. It is when your serum or the patient's serum would say that he is exposed, which means the body responded by production of antibodies. The seroconversion window period specifically refers to the interval between HIV acquisition and the first detection of anti-HIV antibodies. The seroconversion window period varies depending on the different HIV antibody assay used. So the sensitivity will play the major role. So as we see here in this diagram, we can easily see that for the first generation antibody assay, you need at least 35 days. For the second generation antibody assay, it gets shorter, it's more sensitive, it is 30 days. For the third generation, even shorter, 22 days. For the fourth generation, it is even shorter, 17 days. It could be as short as as early as the first detection of P24 antigen in the blood. But as we say, seroconversion is detection of antibodies. Seroconversion window period is first detection of antibodies. And as we said, it depends on the sensitivity of the test. So here, to summarize, the first generation needs 35 days, and of course it continues for more. The second generation 30 days, the third generation 22 days, the fourth generation 17, the fifth generation 17 and more, and the NAT nucleic acid assay is 7 days. Within a week period, we can start at 7 days to detect the virus. The distinct laboratory stage of HIV infection. No HIV test can detect, as we said before, HIV immediately after infection, which we refer to as the eclipse phase. The distinct laboratory stages are referred to. The first marker, as we said, it is the NAT, which will appear within 7 and up to 33 days. And of course, the difference between 7 and 33 is what is the virus load that induced, that was associated with acquisition, and at the same time the immune responses, the presence of ulceration in the genitalia and in other sexually transmitted inflammatory conditions, the transmission of the virus from the exposure site to the regional lymph nodes, where it amplifies through what we referred to before as trans-infection, and the pool of CD4 there that will acquire this infection and their ability to produce the virus progeny. That's why there is a range from 7 to 33. The next marker to appear, to be detected, the P24, usually detected using the antigen antibody immunoassay. There is no antigen assay per se. It's always in combination with antigen antibody assay, and it usually takes from 18 to 45 days after exposure. And as we said, the range is always depending on the host and the virus load, as well as the virulence of the virus. As we said, if it is an F strain that is not replication competent strain, then it will take longer. Time to positive antibody test, again, will depend on the sensitivity of the antibody test, and it will range from 22 as the shortest period to 90 days post-exposure, depending, as we said, to the sensitivity of the test. The CDC recommendation for laboratory HIV testing, the algorithm, will only start by testing utilizing the antigen antibody assay. If it is positive, we have to complete and to identify. If it is negative, then we do not need to proceed, because we are using antigen antibody immunoassay. It is sensitive assay. So actually, if it is negative, it's okay. But if it is positive, we can proceed for differentiation immunoassay. We use the HIV-1, HIV-2 assay and detect. If it is positive HIV-1, then it is HIV-1. If it is positive HIV-2, it's HIV-2. But this is what to identify what type. It is important for the prognosis. As we said, HIV-1 is more virulent. It goes more to severe infection and full-blown AIDS, and requires better and appropriate handling. HIV-2 is better prognostically, but again, without treatment, the patient will progress to disease and will ultimately die from AIDS. If it is positive for both, it could be also positive for both. There is double infection. Or it could be negative or indeterminate. If it is negative or indeterminate, the recommendation is to proceed and do nucleic acid test. If it is negative, it's okay. If it is positive, then this is an acute HIV infection. So this is the recommended algorithm. Starting with an antigen antibody assay. If it is completely negative, then we forget about it. If it is positive, we can do differential assay to identify what type of virus, because if it is type 1, it is a worse prognosis. Type 2, better diagnosis. Or if it is a concomitant infection with type 1 and 2. If it is the result of this test that was positive is indeterminate or negative, then we have to move on to nucleic acid test. Why? Because it could be either true negative, or it could be an acute HIV infection. Now, it is very important to highlight that the Western blot assay that acquired lots of attention previously is now not recommended to be done, because actually it gives lots of indefinite results. So actually it is no more recommended in the algorithm for HIV laboratory identification. I hope that I covered the topics, and any questions by any of the students, please, you can forward them to my email, and thank you very much for your attention.